Hello, I'm Dr. Jeffrey Hayes, Education Specialist for the American Society for Reproductive Medicine. Welcome to the latest presentation of the ASRM Grand Rounds webinar series. These webinars are designed to address topics in the ABOG Guide to Learning in Reproductive Endocrinology and Infertility. Today's presentation will be given by Dr. Catherine Racowsky. Dr. Racowsky is currently in the Department of Obstetrics and Gynecology at Brigham and Women's Hospital at Harvard Medical School in Boston, Massachusetts. The title of her talk today is Early Development, Culture, and Transfer of the Embryo, from Gametes to Blastocyst. I will now review the details of today's presentation. After the webinar is done, please do not forget to return to ASRM eLearn to take the post-test and complete the survey for your continuing education credits. You must complete the post-test question successfully and complete the survey to receive credit and be able to print off your certificate. If you wish to ask a question to the speaker about this presentation, when you return to ASRM eLearn, click on the page link labeled Questions, and an email address to us will be provided. The question period will only be open for a 3-week period after this presentation is posted. After the time period for questions has expired, the questions page will become a frequently asked questions page pertaining to this presentation and topic. We're very excited for our talk today, so I will now turn things over to Dr. Racowsky.
Well, thank you very much, Dr. Hayes. It's a pleasure to participate in the ASRM Grand Round series. So as you mentioned, the title of this presentation is Early Development, Culture, and Transfer of the Embryo from Gametes to Blastocyst. So this slide shows my disclosures. So by way of introduction, as you are probably aware, the first IVF baby, Louise Brown, was born nearly 40 years ago. What you may not know, however, is that it actually took more than 100 attempts before this very first success. This very low efficiency was due to challenges in harvesting the eggs, availability of only 1 or 2 eggs, because of course in those days there was not ovarian stimulation, limited understanding of gamete quality, fertilization and development of the pre-implantation embryo, and also indeed suboptimal culture conditions. So needless to say, over the course of the nearly 4 decades, we've made tremendous strides in all these areas, many of which we will review in this lecture. So at the conclusion of this presentation, the hope is that participants should be able to define good quality gametes, outline critical events of fertilization and milestones in pre-implantation development, review the basic requirements for culturing human gametes and embryos to the blastocyst stage, and discuss the pros and cons of blastocyst versus cleavage stage transfer. So let's now immediately discuss gamete quality. So gametogenesis is dependent on somatic cells, which of course in the male involves the Sertoli cells as well as the Leydig cells. While in the female, these somatic cells comprise the theca cells and the granulosa cells. The granulosa cells fall into 2 subtypes: the membrana granulosa cells, which underlie the basement membrane, which separates the theca cells from the granulosa cell layer, and the cumulus cells, which actually surround the oocyte. The innermost layers of cumulus cells surrounding the oocyte are known as the corona radiata cells. What is of note, though, is that in the female, in addition to the strict morphological relationship between the somatic cells and the oocyte, there are these granulosa cell processes that extend from the innermost layer of corona radiata cells down across the zona pellucida through the perivitellin space to abut on the egg membrane, which is otherwise known as the oolemma. At the point where these processes abut the oolemma, there are gap junctions. Now, gap junctions are channels of communication that allow molecules of about 1 to 2 kilodaltons to go through. So these gap junctions then are extremely important in allowing the communication between the somatic cells and the oocyte. I should say that it is not just between the corona radiata cells and the oocyte that these gap junctions exist, but they also exist among the cumulus cells and also between the cumulus cells and also the membrana granulosa cells. So we have then a functional syncytium in the follicle, which allows communication between the somatic cells and the oocyte. The health of the granulosa cells is critically important for the viability of the oocyte. And by the way, there is crosstalk, so the communication is not unidirectional, but it is actually bidirectional. So there is communication both from the cumulus cells via the corona radiata cells and these processes down onto the egg membrane, but there is also communication in the other direction from the oocyte back to the cumulus granulosa cell layer. So then we have then an oocyte cumulus complex that is attached to the membrane of granulosa cell layers, which comprise a functional syncytium with transzonal processes enabling this bidirectional communication between the oocyte and cumulus cells via these gap junctions. So as we think about assessment of gamete quality, I think we can define it in the following way. We can define it by the ability of gametes to complete meiosis, undergo successful fertilization, sustain embryonic and fetal development, and then finally, of course, to support the birth of a healthy offspring. There are various markers that are used for assessment of gamete quality. So let's, on the left-hand side, first of all, briefly review those for oocyte quality, and then we'll review those for sperm quality. So for oocyte quality, critically importantly, we think about the meiotic stage of the egg. We also critically importantly think about the ploidy of the oocyte, and we'll be talking more about these shortly. We also need to be concerned with cytoplasmic maturity of the egg, gene expression within the oocyte, methylation, demethylation, particularly during imprinting, during very early stages of oocyte development. And then also, as I mentioned, because of the importance of the somatic cells, we can also think about markers of granulosa cell function, which might reflect the overall quality of the egg. So in contrast to these markers of gamete quality where the egg is concerned, for the sperm, again, we need to be concerned about meiosis and ploidy. But of course, we also have various standard conditions or characteristics of the sperm within the semen, such as the concentration, motility, and morphology of the sperm, which we use to appraise the overall quality. Also, there is work that has investigated the relationship between DNA integrity and sperm quality, as well also as gene expression, and again, methylation, demethylation of the gamete. So together then, as I mentioned, in order to be able to achieve a healthy birth, we need to have oocytes and sperm of high quality, which then have and can express fertilization potential, support embryo development and implantation, and sustain a pregnancy, which then altogether will lead, hopefully, to a healthy offspring. Now, I think in the context of gamete quality, one of the things that, as I mentioned, is critically important is this process of meiosis. And I thought it would be helpful to give a brief refresher on meiosis because it is a little bit complicated. So immediately before entry of our stem cells, which are diploid, into meiosis, and of course these stem cells in the female will be the oogonia and in the male will be the spermatogonia, each chromosome in the stem cells undergoes DNA replication. The resulting cells then each will have 4 copies of each chromosome. So here you can see on the right-hand side where N actually refers to the number of sets of chromosomes, and C actually refers to the number of copies of a chromosome, in other words, the number of chromatids that a chromosome has. So the stem cells, before they enter meiosis, they are at 2N, they're diploid with 2 copies of DNA. They then undergo DNA replication. We still have 2 copies of chromosomes, so the cell is still diploid, but there are 4 copies of the chromosomes, in other words, 4 chromatids. So as meiosis gets underway, it consists of 2 divisions. Meiosis 1 involves pairing, synapsis, recombination, and segregation of homologous chromosomes. By homologs, what I'm referring to is the pairs of chromosomes, one member of which will have come from the mother and the other from the father. So the resulting 2 cells in this case then are haploid. So we have then 1N, because now we only have one set of chromosomes, and we have 2C, 2 copies of chromatids. And so there'll be 2 of these. So you can see that 1N, 2C times 2 equates to our 2N, 4C. So after meiosis 1, in meiosis 2, there is separation and segregation of the chromatids. The resulting 4 cells are each haploid with a single copy, in other words, a chromatid of each chromosome. So now you can see that we've gone from haploidy with 2 copies of chromatids times 2 to 4 cells, each of which comprise 1N, 1C. They're haploid with a single chromatid. So now, if this process occurs normally, then we will end up then with, on the one hand, a mature egg that has 23 chromosomes in the human, which is comprised of 1N, 1C. It's haploid with 1 chromosome represented by a single chromatid, and also a mature sperm, also 23 chromosomes, if everything has gone normally, comprised of 1N, 1C. So that when the mature egg and the mature sperm come together at fertilization, they give rise then to an embryo, which then consists of 46 chromosomes, is diploid with 2C, 2 copies. So then what we refer to then is restoration of the diploid complement after fertilization. So it's a complicated process, but very critically, meiosis is essential for sexual reproduction because it allows for this recombination of DNA material between the mother and the father chromosomes, which gives us our genetic variation, which is the whole basis of sexual reproduction. So when we think about the egg, typically in an IVF lab, we grade oocyte maturity typically by the immaturity as either immature or mature. So when we think of a mature egg in the IVF lab, we think of an egg which is at metaphase 2. And this is, of course, reflected by the presence of the first polar body. If the egg does not have the first polar body, we classify it as immature, but actually it can be further classified. And in the IVF lab, we will then refer to eggs that are immature that have the germinal vesicle present, as I'm indicating here, in other words, the GV, as being at the GV stage, which actually also is the same stage as prophase 1 of meiosis 1. If there is no germinal vesicle and there is no polar body present, typically in the IVF lab, we classify the egg as being at metaphase 1. Where you can see there's no germinal vesicle here and there's no first polar body present. However, actually it's not terribly accurate to classify an egg as M1 because the chromosomes, and here you see on the bottom panel the chromosomes, which I'll go through in a little more detail in a second, but the chromosomes actually might have already started to move off the metaphase plate to anaphase 1 or to telophase 1. So let's just look at the bottom here and remind ourselves of what we're thinking about and looking at. So here then, these are fluorescent confocal microscopy images that actually were taken by my colleague, Dr. Catherine Combell, and we published this work in several papers actually. But in any event, this image here is of a germinal vesicle stage egg, and here you see the chromatin labeled in red of the nucleus. And you can see that there is individual little compacted or condensed pieces of chromatin here. But as meiotic maturation gets underway, the chromosomes or the chromatin decondenses and then condenses to form very discrete chromosomes visualized here, and they line up on this little structure in the egg known as the meiotic spindle, and these green areas or fibers here actually represent the spindle fibers, and the chromosomes have paired up at metaphase 1 on the metaphase plate. Shortly thereafter, the homologs, the paired chromosomes, then segregate. So here is the metaphase plate here at anaphase 1, and here are 2 groups of segregated chromosomes, one of which then will eventually be emitted from the egg at telophase 1 to result in the first polar body. And here you can see this process of telophase 1 actually occurring, where here is the meiotic spindle. The chromosomes that are retained in the egg are shown here. The yellow arrow is pointing to the chromosomes that get emitted into the first polar body. And here, by contrast, the first polar body, I'm sorry, the image is a little switched, but here is the DNA indicated in red in the first polar body, and it's actually undergoing degeneration. And here we see the second meiotic spindle with the chromosomes lined up on the second metaphase plate. And although the magnification here is lower than it is here, hopefully you can see just by judging the relationships that you'll see there's about half the amount of chromatin here on the second meiotic plate or metaphase plate as compared with on the first metaphase plate, which is what one would expect, given that I've just said that we get a reduction of the amount of chromatin material as we go from meiosis 1 to meiosis 2. So of note, this process of meiotic maturation, going from the germinal vesicle stage to metaphase 2, actually takes around 36 hours. And believe it or not, the founder of IVF, from the basic science point of view, Dr. Robert Edwards, actually documented this process of going from the germinal vesicle stage to metaphase 2 as taking 36 hours, and he did this in 1965, which is over 50 years ago. So we've known this for a long time, but this is why when we ask our patients to take HCG, they have their retrieval 36 hours later, because we expect this process of meiotic maturation to go from the germinal vesicle stage to metaphase 2 over this 36-hour period. So now there are many, many unfortunate problems that might occur during meiosis. Some images shown here are representative examples of some of the problems. So here we see a normal metaphase 1 spindle again. Here's our spindle with our chromosomes nicely lined up on the metaphase plate. But you can have situations where you see a grossly abnormal meiotic spindle here, where you can see the chromosomes or pieces of chromosomes are very dramatically and randomly dispersed among these spindles, and they're actually, you can see that actually it's not just a bipolar spindle with two poles, but you can see there are multiple spindles here. And in addition to that, you can have abnormally shaped spindles, such as shown here, and where, because of the abnormality, one of the reasons might be for abnormal distribution of the chromosomes and disorganization of the chromosomes among the spindle. And conversely, you might have a reasonably shaped normal spindle, but nevertheless abnormal chromosome dispersion with disorganization among the spindle fibers themselves. So we are all aware, I think, of the problem of aneuploidy and how this relates to female age. This work was published by Jason Franasiak from Richard Scott's group in 2014, and what it shows here is the relationship between maternal age on the x-axis and the incidence of aneuploid embryos on the y-axis. And as you can see, interestingly, it is actually sort of an interesting sigmoidal-shaped curve in that the lowest incidence of aneuploidy appears to occur around 28 to 29 years old. But you can see that there is a dramatic rise in the incidence of aneuploidy as maternal age increases, particularly after the age of 35. Now, what you may not appreciate as well is that actually around the age of 36, we can expect on average around 35% of embryos to be aneuploid based on this work, which, by the way, involved a very, very large number of embryos. But in contrast to the age of 36, just 5 years later, at the age of 41, we, according to this work, can expect a doubling in the aneuploidy rate from 35% to 70%. So aneuploidy is clearly a big problem in clinical IVF and actually also in human reproduction. Now, in addition to nuclear maturation, as I mentioned earlier, we also have to think about cytoplasmic maturation of the egg. So again, nuclear maturation involves the accurate chromosome alignment and segregation and formation of an oocyte with 23 chromosomes, whereas cytoplasmic maturation involves the support of nuclear maturation sperm penetration, syngamy, the joining together of the two pronuclei after fertilization, and then the support of embryo development. So together we think of nuclear maturation and cytoplasmic maturation as resulting in acquisition of developmental competence. And if an oocyte has not undergone both nuclear maturation and cytoplasmic maturation, then it will not be able to support embryo development and a healthy pregnancy. So the acquisition of developmental competence with respect to cytoplasmic maturity certainly involves some discrete features. So as the egg is undergoing development, it is, of course, initially nuclearly and also cytoplasmically immature, and is characterized by being small in diameter, around 80 microns in diameter. It exhibits dispersed chromatin within the nucleus, and the mitochondria are dispersed within the cytoplasm, and the egg itself is high in RNA synthesis. As development proceeds and the egg is acquiring developmental competence, it becomes nuclearly competent in that the chromatin assumes a ring-shaped configuration, and RNA synthesis ceases. The egg also proceeds in growth and assumes pretty much its maximum growth to around 110 microns in diameter. Notice, however, that the mitochondrial aggregates still remain dispersed within the cytoplasm. Finally, with complete acquisition of developmental competence, the nuclear chromatin remains in its ring-like configuration, but you'll note that the mitochondria are now no longer dispersed within the cytoplasm of the egg, but actually now aggregated towards the nucleus of the egg itself. Again, the fully developmentally competent oocyte is of the order of around 110 microns in diameter, and again, RNA synthesis has been shut off completely at this stage. So as we think about the quality of the gametes, I think it's important that we consider the ovarian, the sort of principles underlying ovarian stimulation, because not all eggs, as any embryologist who is listening to this grand rounds will appreciate, not all eggs are certainly mature, and there's a large variation in any cohort of eggs that's retrieved. So let's just review what happens, first of all, during a normal menstrual cycle. So you can see here that we have a group of growing follicles, so a cohort of follicles that has been recruited to undergo growth in perhaps 2 or 3 months prior to this index cycle. And this group of follicles will consist of any number depending upon the age of the woman and her ovarian reserve. In a young woman, we might expect somewhere of the order of 30 to 50 growing follicles. And then around days 3 to 5 of the menstrual cycle, within this group of growing follicles, one and only one of those follicles will then become selected to become the dominant follicle. And it will continue to grow, ultimately to be the one that is ovulated if the woman is allowed to undergo a natural ovulation in the cycle. All the other follicles in this growing cohort undergo degeneration, in other words, this process that we refer to as atresia. So then what are we doing when we actually do ovarian stimulation? Well, what we're doing, of course, is we're adding exogenous gonadotropins, the follicle-stimulating hormone, with or without human menopausal gonadotropin. And what we're doing is we're rescuing or attempting to rescue all these follicles that would otherwise undergo degeneration and bring them back on the pathway of growth, so that when we actually trigger our patient with HCG, human chorionic gonadotropin, we hope then that all of these follicles containing eggs will respond appropriately to this gonadotropin, and the eggs within these follicles will behave like the dominant follicle, so that we end up with a large number of meiotically mature eggs from this stimulation regimen. Remembering that from the time of the HCG injection, which we refer to as time 0, the retrieval should occur around 36 hours later for the reasons that I just stated, that this 36-hour interval is the time required on average for the egg to undergo nuclear maturation. So as I mentioned, we all know that generally speaking, a cohort of eggs that's retrieved is heterogeneous in characteristics. And indeed, only about 75% of eggs are mature. Not all mature eggs are euploid. Not all euploid oocytes fertilize and develop normally. And so there is therefore considerable oocyte and embryo wastage in clinical IVF. The biological efficiency of the oocyte to the baby has been estimated by Patricio and Sackers, who published a paper in 2009, to be only of the order of around 5%. So you can see that the overall efficiency of clinical IVF is relatively inefficient. It may be, though, and I would just say as a caveat, that in the last 5 to 6 years or so, 5 to 7 years, as we continually improve our technologies, this efficiency may have gone up marginally, but it still nevertheless is very low. So if you think about it, even if we assume that the efficiency is 10%, that means that only 1 out of every 10 eggs that we retrieve actually will end up giving rise to a live birth. So we handle in the IVF lab, unfortunately, a lot of abnormal oocytes which have no opportunity to ever develop into a healthy pregnancy and a live born. So the key points for gamete quality are then, first of all, that gametogenesis is dependent upon somatic cell control. As I mentioned, the Sertoli cells, particularly spermatogenesis, as well as the Leydig cells, and as far as oogenesis is concerned, particularly the granulosa cells, as well as to some degree the theca cells, due to the stereogenic control of the whole process. Quality of the gametes are defined by the ability to support development of a normal, healthy birth. And although ploidy is a key marker of quality, cytoplasmic maturation of oocytes is also absolutely required. The quality of the oocytes in a retrieved cohort after ovarian stimulation is typically variable, and as I mentioned, the overall biological efficiency is very low. Okay, so let's now discuss fertilization and milestones in pre-implantation embryo development. I will also be giving a brief discussion on time-lapse imaging. So what are the critical events during fertilization? Well, first of all, obviously, we have to have binding of the sperm to the zona pellucida. This actually involves sperm binding to the third layer, the ZP3 layer of the zona. In the human, there are actually 4 glycoprotein layers to the zona pellucida. So the sperm binds to ZP3-1 after penetration of it binds to ZP3 after penetration of ZP1 and ZP2. And this binding involves the sperm binding protein, which at the moment at least we believe to be 1,4-galactosidase. After binding of the sperm to the zona pellucida, the acrosomal reaction is initiated, and this, of course, involves the release of enzymes from the acrosomal cap from the sperm head, which then allows penetration of the sperm through the zona pellucida. Following penetration through the zona pellucida, the sperm then binds to the oolemma, and this sperm binding is elicited by the sperm protein izumo-1 and the cognate oolemma receptor Juno, which actually together results in depolarization of the oolemma and cortical granule release. So this is a very important event during fertilization. So after this occurs, there is fusion of the plasma membranes, the oolemma, with the plasma membrane of the sperm head, and the sperm nucleus then enters the oocyte, or the ooplasm. So after fusion, the sperm-derived phospholipase C-zeta mobilizes intracellular calcium stores within the oocyte to induce oocyte activation. And it's oocyte activation then which then triggers the cascade of events which ultimately give rise to pronuclear formation and then fusion of these pronuclei at syngamy. So in the IVF lab, typically then we will examine the oocyte for fertilization around 16 to 18 hours after we've done either standard insemination or our ICSI procedure. And what we're looking for at the fertilization check is the presence of 2 pronuclei and only 2, one of which will have formed from the male chromosomes, from the sperm chromatin, and the other from the female chromosomes. We believe, generally speaking, the larger of the two pronuclei is that derived from the sperm. And you can see here that within the pronucleus, there are these little structures, the nucleolate precursor bodies here, which is the RNA making or manufacturing, synthesizing machinery of the nuclei. And you can see the presence of the two polar bodies, the first one of which would have been released at telophase 1, so telophase is the first meiotic division. The second one, which is slightly out of focus here, would have been released after the sperm has penetrated the egg, when the egg completes the second meiotic division and goes from metaphase 2 to telophase 2. So in contrast to visualization of two pronuclei, sometimes we only see one pronucleus. We may see three pronuclei, or alternatively, we may see more than three. Generally speaking, if we see three pronuclei, particularly if only one polar body is present, this is caused by failure of emission of the second polar body, although it could also be due to disperny, where two sperm have actually entered the oocyte. So it's very important during the fertilization check that we check very carefully that there are only two pronuclei present in each zygote that we deem to be normally fertilized. So after we've done the fertilization check, then we will place the embryos back into culture, and the embryos then will proceed through development, as shown in this slide. So our newly formed zygote, here it is, with our two pronuclei shown here, will then, around 24 to 27 hours after insemination or ICSI, will cleave to form the two-cell stage late on day 1, which then will cleave to form the 4-cell stage on day 2, and then ultimately will cleave again, if everything is developing normally, to form the 8-cell stage on day 3. There is then a further cleavage division to at least the 16-cell stage before the embryo undergoes this process called compaction on day 4. And in compaction, what we're referring to here is the formation of tight junctions between the cell membranes of the blastomeres, and the loss of ability to actually be able to visualize clearly the beginning of each cell and the end of each cell. So it appears much more sort of like a block. This process of compaction is critically important because it allows the accumulation of tiny little pockets of fluid, and actually I think here you can see a very small pocket of fluid beginning to form, as possibly here and here. These pockets of fluid ultimately coalesce to form the so-called blastocelic cavity on day 5. And the other very important thing to note about the blastocyst, which I'm sure you appreciate, is that this is the stage at which we see overt differentiation of the two cell lineages, the trophectoderm cell lineage, which goes to form the embryonic part of the placenta, and the inner cell mass lineage, which goes to form the fetus itself. And of course, this is where stem cells are derived. These are pluripotent cells. So as the embryo proceeds in development, the blastocyst continues to expand, and ultimately it will hatch from the zona pellucida. You can see how the zona pellucida actually decreases in thickness as development proceeds. And here you see a blastocyst streaming out of the zona pellucida as it's undergoing the hatching reaction. The implantation in the human we believe to occur somewhere between day 6.5 and day 7. And of course, the embryo has to be completely hatched from the zona pellucida in order for implantation to proceed normally. So the embryo normally progresses along a strict developmental timeline. And it was actually Sir Robert Edwards who was the first to critically define this developmental timeline in the human embryo way back in 1981. Here he described the kinetics of development from the 2-cell stage through to the 16-cell stage. But during his observations, he noted that not all embryos proceed along this normal developmental timeline, with some developing more slowly and others developing more rapidly. However, now, from a number of investigations, we know that there is a fairly substantial percentage of embryos that do indeed develop abnormally. For example, an embryo may take longer than normal to reach the 2-cell stage, then be slowed up in development, but then proceed more rapidly from the 8-cell stage to the 16-cell stage, so reaching the 16-cell stage more or less at the normal timeline. Alternatively, an embryo may enter the first cleavage division more rapidly and proceed more rapidly through to the 8-cell stage, but then become delayed in development, but nevertheless reaching the 16-cell stage at more or less the normal time. And finally, some embryos may initiate cleavage more slowly, but nevertheless follow a linear progression so that they ultimately reach the 16-cell stage, but they do so at a different time period. And conversely, some will also proceed along a linear progression, as shown here by the brown line, but having started cleavage much earlier than normal, they will reach the 16-cell stage much earlier than normal. So key points with respect to fertilization and embryo development are as follows. Fertilization involves critical events, including binding of the sperm to zona pellucida 3, the third layer of the zona pellucida, which initiates the acrosome reaction. Interaction between sperm protein isomo-1 and its cognate receptor Juno on the O-lemma occurs, and after sperm-O-lemma fusion, the sperm-derived phospholipase C zeta mobilizes intracellular calcium stores to induce oocyte activation. Competent human embryos, importantly, develop along a predictable developmental timeline, normally, and certainly not all embryos progress normally in vitro, and presumably the same is the case also in vivo. So because of the abnormalities that we have observed over time, we are also interested in understanding not just how the embryo might appear during conventional morphological evaluation, but also at much more frequent intervals of time. And during the last, 7-8 years or so, time-lapse imaging has really again been reintroduced into clinical IVF, where we can actually image the embryos perhaps every few minutes and get a very substantial library of information as to how the embryos are developing over time. So with this time-lapse imaging approach, we've been able to much better define embryo morphokinetics. And various markers of morphokinetics have been developed, such, for example, as the time to the 2-cell stage, time to the 3-cell stage, time to the 5-cell stage, time to the 8-cell stage, morula, early blastocyst, and expanded blastocyst. And there have been discrete windows that have been defined that would typically represent an embryo as it proceeds through normal development. In addition to defining these embryo morphokinetic markers, also abnormal phenotypes have been observed using time-lapse imaging. And this slide shows you 5 different abnormal phenotypes. Abnormal syngamy, where rather than the pronuclei coming together in a very smooth fashion toward the center of the egg and then undergoing syngamy, they tend to spin around before they actually join together. The second abnormal phenotype is abnormal first cytokinesis, and this is where one can see ruffling of the O-lemma, which is abnormal. Typically it is smooth as the egg, the newly formed zygote, cleaves from the 1-cell stage to the 2-cell stage. Thirdly, there's chaotic cleavage, where you get literally just complete lack of control of cell division, and the zygote exhibits as really a ball of fragments. And then fourthly, there are 2 different subtypes of abnormal cleavage, where you can have abnormalities of the first cleavage and abnormalities beyond the first cleavage. So with first cleavage, we can observe abnormal cleavage where the zygote develops from the 1-cell stage directly to the 3-cell stage and doesn't go to the 2-cell stage at all. And in abnormal cleavage 2, a daughter cell cleaves yielding more than 2 blastomeres, so that rather than 2-cell stage cleaving and forming a 3-cell stage first, which is the normal progression, you observe the 2-cell stage forming an embryo that actually has 4 cells. 1 is much larger and 3 much smaller. And so ultimately, in this situation, you might get an embryo that might then form 5 cells or more. And alternatively, as you can see here, you can get a situation where the 3-cell can go to the 5-cell stage. So these abnormal phenotypes have been correlated with implantation potential. This work was published by Kelly Worker in 2014. And as you can see here, each one of these pairs of graphs describes the implantation rate of a particular abnormal phenotype. So here is abnormal syngamy, and you can see, although the numbers of zygotes that have been actually analyzed and embryos that have been analyzed is still relatively small, you can see that those embryos that exhibit abnormal syngamy have a very low, I think just above 0% implantation rate. And likewise with abnormal cytokinesis 1, with abnormal cleavage, and also with chaotic cleavage, you can see that the implantation rates are substantially reduced for those embryos that exhibit these abnormal phenotypes. So as I mentioned earlier, various morphokinetic markers have been identified which describe the time intervals through which the embryo develops during pre-implantation development. And using these markers, various tracking algorithms for embryo selection have been developed. The first of which was developed by Gamete, and he published the comprehensive algorithm in 2012. And then the other algorithm, which is known as the EVA test, was published by Conahan et al. In 2013. So in both instances, the timing of development of the embryo is analyzed, and there's a deselection of embryos as the embryo goes through development, depending upon whether the embryo falls within the defined time period which was considered to be normal. You can see that the EVA test is a much simpler algorithm as compared with the Gamete algorithm. All of the algorithms to date have been designed to be used in conjunction with embryo morphology, which is an important point. So there's a growing literature investigating the use of time-lapse imaging versus conventional morphology for embryo selection. Importantly, the majority of these studies are retrospective, and mixed results have actually been reported. And this has been nicely reviewed in a very recent article by Costello et al. Published last year. So what does the evidence tell us from the randomized controlled trials? Well, before we actually look at the evidence, I think it's of use to actually think about the actual time-lapse imaging system and what it consists of. So there are actually two components to the time-lapse imaging system. There is the incubator, and there's the actual selection method. So with the EmbryoScope and the Jerry, for example, which are just two of these more compact time-lapse imaging incubators on the market, you can see that the incubator and the time-lapse selection system itself, or the algorithms, are actually, the computer system is actually built into the same unit. In contrast, for the PrimoVision and the EVA test system, the microscopes themselves actually do the video recording, but these are placed within standard box incubators. And this is the case, I say, both for the PrimoVision and also for the EVA system. So, and this, of course, is the typical box incubator system, whether it's a bench incubator or a normal tissue culture incubator, is the way that we normally culture embryos. So when we think about the design of randomized controlled trials, then we need to take into consideration both the incubator that was used as well as the selection method that was used when comparing the time-lapse imaging arm with the control arm. So let's just look at the three possible paradigms for the time-lapse imaging system for the randomized controlled trials, of which there are 5 to date. So you can have, in the first instance for the time-lapse imaging system, you can have the time-lapse incubator with time-lapse selection, and this can be compared to our control system, which might consist of the standard incubator and standard selection. If you compare these two things, because the control system consists of the standard incubator and standard selection, we're only actually comparing the system here. Conversely, you can have for the time-lapse system the time-lapse incubator and time-lapse selection, which is then compared to a time-lapse incubator in which the embryos are selected using standard selection, in which case you are actually testing the selection method itself. And finally, you can have the paradigm with the time-lapse incubator and standard selection, and the standard incubation and standard selection, in which case you're testing the incubator. So as I mentioned, 5 randomized controlled trials have been done to date. With respect to the system, there are 3 randomized controlled trials for selection, there are 2, and for the incubator, to my knowledge, there are currently none. So what does the data actually show us with these randomized controlled trials? So this slide shows you the 5 randomized controlled trials here. For this particular study, the Rubio study, I've separated the transfers into day 5 and also day 3, as also for the Goodman study. So you can see here the % ongoing pregnancy rate per embryo transfer when the embryo is selected after culture in the time-lapse system versus in the standard system. And you can see that actually for all but one of these comparisons, there was no difference between the ongoing pregnancy rate between these 2 paradigms. The only time at which there was a significant difference was in the Rubio study for those embryos that were transferred on day 5. So myself and others have done meta-analyses to try to understand, when collating all this data together, what the actual final analysis is with the available data for using time-lapse selection for embryo evaluation. And so this slide shows a forest plot for the randomized controlled trials for ongoing pregnancy. It's actually a modification of a paper we published in 2015. But what you can see here is the relative risk for time-lapse imaging versus control is 1.11 in favor of time-lapse imaging, but however, there's not a significant difference because the confidence intervals do cross 1. So key points for time-lapse imaging and embryo selection are as follows. Abnormal, unique phenotypes are associated with lower implantation rates. There's more work that needs to be done in that regard for sure, but the current data is compelling. Tracking algorithms with time-lapse imaging have been identified. Despite a large number of retrospective studies, some of which have shown that time-lapse imaging markers are associated with blastocyst formation, the few prospective studies collectively fail to demonstrate improvement in implantation rates with time-lapse selection versus standard morphological selection, and most of these randomized controlled trials are of low quality due to design issues of one sort and another. So I think it's fair to say that for sure more well-designed randomized controlled trials are required before we can say definitively that time-lapse imaging actually does enhance our ability to select the best embryo to transfer or embryos to transfer compared with standard morphological selection. So now let's turn to requirements for culturing human gametes and embryos to the blastocyst stage. So why is there a concern about this at all? Well, I think in order to answer this question, we can just spend a few minutes thinking about the differences between the in vivo environment and the in vitro environment. So in the in vivo environment, the environment is three-dimensional. It is certainly dynamic. Please remember that, as I mentioned earlier, the diameter of human egg and indeed embryo until it undergoes expansion at the blastocyst stage is of the order of around 110, 115 microns. It is tiny. And in vivo, it traverses the tube down into the uterus, not really in a fluid at all. It's much more in a polysaccharide matrix where the microvilli in the fallopian tube actually nest the embryo as it's moving down the tube. In contrast, the in vitro environment is two-dimensional, it is static, it is fluid, by the way, and it also poses multiple challenges. It poses challenges with respect to temperature control, light control, pH of the media in which the eggs and the embryos are cultured, the osmolarity of the medium, the constituents of the culture media, and also the other contact materials, of which there are a very large number in human IVF, remembering that in vivo, the egg and the embryo does not come into contact with any of these artificial materials. And then finally, there's the complexity introduced by all the various manipulations that we perform on the eggs and the embryos. And we know from now a large body of work that human gametes and embryos are exquisitely sensitive to their environment, and the IVF culture system is very complex. So the system consists of many different components, as I think we probably all know. The incubator type is important, the type of culture dish, the density of the embryos, whether we culture them singly or in groups, the oxygen tension, and I'll be saying more about that, and also the culture media, of which there are a very large number, or relatively large number, of commercially available types. But because of the complexity of the IVF lab, quality management in the lab is absolutely mandatory in order to ensure safety of the gametes and the embryos. So quality management in an IVF lab consists of many different things. It consists of written policies and procedures, which must be current, validated, and implemented. It consists of managing the equipment, which must be maintained. The performance must be validated through quality control work. I'll say a little bit more about that shortly. Personnel, the embryologists and the andrologists, must be trained to perform the procedures. They must be certified, and indeed there must be constant monitoring of their performance to make sure that there's not personnel drift over time. And then, of course, we can think about the patient and the quality of the ovarian stimulation and the quality of the gametes that are produced, but this doesn't necessarily or really relate specifically to quality management in the IVF lab. And here you can see below the horizontal line here all the different processes and procedures that we perform, including, and I haven't included it, but embryo biopsy as well. So in addition to all these things, we have to be concerned about the environment. We have to be concerned about the contact materials, and we also have to be very much concerned about the quality of the air. Up until perhaps between 5 and 10 years or so ago, air quality was not very well paid attention to when considering the running of an IVF lab. But we now know that air quality and the purity of the air is critically important for maintaining and maximizing developmental competence of the eggs and the embryos that we're culturing. So quality management then really consists of 3 different things: quality control, quality assurance, and then quality improvement. So through quality control and quality assurance, we monitor and verify acceptable lab performance, and we do this through data collection and analysis, as I mentioned, with constant surveillance. Only if we are performing quality control and quality assurance appropriately and our lab is in control, then we can actually do quality improvement work where we can introduce and validate new protocols into the lab. If the lab is not in control and one tries to perform quality improvement activities, the chances are you're going to end up with meaningless data because there will be so much background noise introduced by the lack of control over the lab performance. So what do we mean by quality control? Well, documentation of instrument and material performance, daily recording of all instrumentation, testing of all contact materials with mouse embryo and/or sperm assay, confirming alarm calibrations and reliability is critically important. All instrumentation, particularly incubators and the liquid nitrogen freezing storage doers, should be alarmed. And confirmation of the incubator performance, in other words, the calibration of the instrument with respect to temperature, gas, particularly carbon monoxide, with respect to medium pH, and also osmolarity of the medium. And then documentation of personnel performance. This should be for all lab procedures for which an individual has been trained and certified. And as I mentioned, there should be competency assessments that should be a routine, at least within perhaps every 6 months. Very importantly and critically, with respect to quality control, integrity of patient identification must be maintained. And at least in my lab, we use 2 identifiers. We use the last name of the patient, and we also use a unique case identifier number. So rather than using a medical record number, which is actually a sequence of nonsensical sequence of numbers, we use the case ID, which today is January the 11th, 2017. So it is the date on which the retrieval occurs, or the procedure occurs. It occurs if it's a thaw. and also the order of it. So this would be the first retrieval of the day to day would have this particular case ID number. And we transfer this and use this along with the name on every single vessel or vehicle that contains the patient's gametes and also embryos. So use checklists. Checklists are extremely useful in an IVF lab, and in my view, they're actually really, really important as well. They help confirm the activities that are critically important when performing this work so that no mix-ups occur. If you've not read this book by Atul Gawande, I strongly recommend it. The Checklist Manifesto. It's a very easy read. It takes probably just a few hours to read. And as you go from page to page, you'll just keep saying, well, of course, this all makes sense. So I very much recommend that you read this book if you've not done so. And if you do not have checklists in your IVF lab, I recommend that you introduce them. I'm happy to share our checklist with you if you'd like, and answer any questions, of course, in that regard. So in contrast to quality control, quality assurance involves the analysis. So I always think of, if I'm thinking about definitions of quality control and quality assurance, quality control involves data collection, control collection. Quality assurance involves analysis, assurance, and analysis. So during quality assurance, then you're analyzing the data you've collected through quality control, and you're monitoring the outcomes and you're troubleshooting them. So this monitoring should be done at regular intervals. It's important to pinpoint quantifiable outcome indicators and then undertake corrections and reevaluations as necessary. So as I mentioned, one of the quality indicators that we use in my lab is the fertilization rate, both with respect to the percentage of zygotes that have 2 pronuclei and also the percentage that are 3 pronucleate. And these are expressed of the percentage of mature oocytes, i.e. of those at metaphase 2. And I'm lucky in that I get an automatic dumping every week of these rates, which are plotted for me. And we have established the target for the percent 2PN rate of M2s being at 70%, and that of 5% for the percent 3PN rate. And as you can see, and as would be expected, because we're dealing with biology, there's biological variation, and you can see how the data does move from one week to another. However, if a point falls below the lower confidence interval, such as here, then one has to do work to understand what the underlying reason for that dip below the acceptable target rate was. And this might, of course, be due to patient-specific issues, or it could be due to a lab problem. And the job of a lab director or manager is to ensure that the lab performance is running correctly and that this dip down was not due to lab performance. In this particular case, as you will see, there was an increase in the percent 3PN rate for this particular week. And during the work that we did, we found that this was actually a patient-related phenomenon. There just happened to be a group of patients that were older. On the one hand, where older eggs are more prone to actually form 3 pronuclei, but more importantly, there were a few patients that actually had repeatedly had a high incidence of 3PN rate on previous cycles. So we concluded that this was a patient-specific phenomenon and not due to lab performance. So a second indicator that we use routinely in our own IVF laboratory is the percentage of embryos that have at least 7 cells on day 3. And just as with the P-chart that I just showed you for the fertilization rates, we have a P-chart for the performance for this particular indicator. And as you can see, we've established a target for this performance with an upper and lower confidence interval. And each week, the data are plotted to assess whether the performance of the lab and the data is within the ranges that have been stipulated. If there is a drop in performance, then the lab has the responsibility to do all the work necessary, the quality assurance work, analyzing all the quality control parameters that have been collected in order to ensure that this drop in performance is not related to lab performance, but is more likely related to specific characteristics of the patients that underwent IVF in that week. And if everything has gone well, then in the following week, there should be a recovery in performance, which again should be obviously tracked every week thereafter to ensure that the performance is within the established variables and the lab is performing correctly. So key points regarding requirements for IVF culture. First of all, human gametes and embryos are exquisitely sensitive to their environment. Therefore, an effective quality management program is essential when running an IVF laboratory. Quality control indicators should be used routinely. For example, as I've discussed with you, percent fertilization of both 2PNs and 3PNs are useful, as well as, in our hands at least, percentage of embryos that have formed at least a 7-cell stage on day 3. And by the way, if you're going to use that as a marker, you must make sure that you are evaluating your embryos over a very specific time period, because otherwise you'll introduce noise in the data and make it much more difficult to use this as a marker for quality assurance. With use of these identified markers, there should be active, constant monitoring of lab performance and corrective action as indicated. All quality control measures should be routinely checked to ensure acceptable performance. Okay, so now let's turn our attention to culturing embryos to the Blastocyst stage. And we'll first of all talk about oxygen tension, which has received a fair amount of attention over the last several years. So routinely and historically, animal embryo cultures have typically been performed in what we refer to as low oxygen tension, that's 5% oxygen, in an atmosphere consisting also of 5% CO2 with a balance of nitrogen. The first human IVF birth, Louise Brown, actually arose from an embryo that was cultured in this gas mix of low oxygen tension. But until the last decade or so, many clinical IVF labs used a gas phase of 5% CO2 in air, which has an oxygen tension of around 20%. Recent work has investigated a possible benefit of lowering the percent oxygen for clinical IVF. First of all, down to 5% to reflect that of work done with animal embryos, and then now very, very recently, down even lower to 2% oxygen. So let's first examine all the randomized controlled trials for comparing low oxygen, that's 5% oxygen, to atmospheric oxygen at 20%. And what this forest plot shows you is on the top, transfer of embryos at the cleavage stage. Secondly, transfer of embryos at the blastocyst stage. And thirdly, miscellaneous, where they couldn't easily be grouped into one or the other because of the way that the trials were run and reported. What is of note here is if you look now at the risk ratio and the 95% confidence intervals, you'll see that there's no difference in the birth ongoing pregnancy rate, and here ongoing pregnancy rate was defined as pregnancies to at least 12 weeks. You can see there was no difference in the birth ongoing pregnancy rate when embryos were cultured just to the cleavage stage and then transferred. But conversely, when the embryos were transferred to the blastocyst stage and then transferred, you can see that there is a significant effect of oxygen tension in that it benefits or favors the 5% oxygen as indicated here. Overall, when combining all the data together, you can see a very marginal improvement in the birth ongoing pregnancy rate when embryos are cultured in 5% oxygen as compared with 20% oxygen. So despite this, I think it's very important to note that the evidence supporting the above is of very low quality, and more properly controlled and designed trials of high quality need to be performed before we can definitively state whether 5% oxygen is actually beneficial to 20% oxygen for culturing human embryos beyond the cleavage stage to the blastocyst stage. So as I mentioned, a very recent work has tested the hypothesis that ultra-low oxygen might actually be helpful and beneficial in the development of human embryos. So here then we are referring to comparison of low oxygen at 5% versus this ultra-low at 2%. And the rationale for this study was that work has previously shown that the oviductal oxygen tension in non-human primates was of the order of 5 to 7%, whereas in the uterus, in both primates and human, the actual PO2 or oxygen tension is much lower, of the order of 2%. So the question arises, therefore, as to whether it is beneficial to culture human embryos from day 1 to day 3 in 5%, and then from day 3 to day 5 in 2%, as compared with culturing embryos exclusively in 5%. I should immediately say that this hypothesis was advanced by our previous clinical fellow, Dr. Daniel Kaiser, who's now working in Richard Scott's group in RMNA, New Jersey. So we performed this study to test this hypothesis. And the study involved a sibling zygote randomization in which the embryos were randomized within a woman either to low oxygen tension, that's 5%, or to the 2% arm. They were randomized on day 1, which is important. So here you see the 3 different characteristics that were evaluated first. The percentage of embryos that form early blastocysts, the percent that form advanced blastocysts, and the percent that form usable blastocysts. And in this first left-hand column are the controls, those that were cultured throughout in 5% oxygen, although notably the embryos were transferred, they were taken out and transferred to different media, both in this group and in this group on day 3. So you can see the percentage of early blastocysts in the control group as compared to the experimental group, where the embryos were first cultured in 5% and then transferred from 5% to 2% from day 3 to day 5. And you can see that the adjusted odds ratio 95% confidence intervals are shown here with a p-value of 0.02. Although there was no difference in the percentage of advanced blastocysts formed, overall the percentage of blastocysts that were usable was significantly increased, with just over a 2-fold increased risk with respect to culture in 2% versus 5% oxygen. And Dr. Kaiser also looked at the number of cells in the embryos. So on the y-axis here, you see the mean number of cells. On the x-axis is the stage that the embryo is developing at. And you can see that as the embryo develops to at least the early blastocyst stage and the advanced blastocyst stage, the actual number of cells in the embryo are significantly lower in those embryos that were cultured in 2% oxygen, as indicated by the red line here, as compared to 5% oxygen, indicated by the gray line. So key points for oxygen tension and embryo culture are as follows. The available data shows, with respect to the 5% versus 20% paradigms, a small improvement, about 5%, in live birth ongoing pregnancy rate following culture in 5% oxygen for blastocysts, but not cleavage stage transfer. Notably, however, the data are of very low quality. With respect to the one study to date that has investigated 5% oxygen and then 5% oxygen versus 2% oxygen from day 3 to 5, that we observed an increase in the percent of usable blastocysts after culture in the 2% oxygen from days 3 to 5, which was accompanied by a total decrease in cell number. Notably, these data are preliminary, and the study was performed with heterogeneous samples. I didn't mention that the majority of the embryos that we used in this study, of course, all of which were donated, were actually 3 pronucleate. So I think it's fair to conclude then, and a key point would be regarding oxygen tension embryo culture, is that more well-conducted randomized controlled trials are needed to resolve the optimum percent oxygen required for culturing human embryos. So now let's turn our attention to culturing the embryo to the blastocyst stage and say a few words about culture media. So in our field, there are basically 2 different paradigms for culturing embryos to the blastocyst stage with respect to media. First of all, the sequential media system, and then secondly, the single-step media. So the sequential media system involves culturing the embryo in media 1 from day 1 to 3, and this media, among other things, including amino acids, is different from the media 2. It has low glucose, whereas the media 2, which involves culturing the embryos from day 3 to day 5, has high glucose. And as I mentioned, also other constituents are different, including amino acids. But the point here is that it is a sequential media system involving two different media types of different constituents. And these media were designed particularly and initially by David Gardner from work that he had done where he had analyzed the constituents of fallopian tube and uterine fluid, although as I mentioned, these aren't really fluids, and which he had shown particularly that there were differences in energy substrate concentration in the two different portions of the human reproductive tract, in that glucose was lower in the fallopian tube than in the uterus, and it was also higher in lactate in the tube versus the uterus. So that's the premise of the sequential media system. In contrast, the single-step media involves, needless to say, a single media in which the embryos are cultured from day 1 to day 5. In some labs, if a single media is used, the media is refreshed on day 3, but the same media is used. In other labs, the embryos are cultured continuously in the same drop from day 1 to day 5. So what does the actual data tell us with respect to trials that have been undertaken where comparing sequential versus single-step media for culturing embryos at the Blastocyst stage? Well, there have been two approaches that have been used for these studies. The first, the patients themselves have been randomized, so all the embryos from one patient were cultured in one media system or the other. And then, of course, one can then compare the clinical outcomes. Two trials were involved in this regard. Alternatively, rather than randomizing the patients, the embryos themselves were randomized within a patient so that half her embryos were cultured in single media and half were cultured in the sequential media system. In this case, the overall blastocyst formation rate was compared, and there were 10 trials involved, and also the percentage of high-quality blastocysts was compared, in which case there were 6 trials. So these are the data showing the results for the randomized controlled trials using these two different approaches. So with respect to clinical pregnancy rate and live birth rate, as you can see, absolutely no difference in the adjusted odds ratio for the two different paradigms, the single media versus the sequential media. And likewise, with respect to percent blastocyst and percent high-quality blastocyst, really no difference, except one might say there's a very slight trend towards improved blastocyst formation rate in the single media, 50.3% versus 48.7%. And note, there are a very large number of embryos here, and the confidence interval at the lower end does not cross 1. So in principle, this is a significant effect, but it's a slight trend only, and in clinical terms, an improvement of about only a 2% increased blastocyst formation rate. So key points regarding media for culturing blastocysts are, first of all, no difference in clinical outcomes for sequential versus single media. There was a slight trend towards improved blastocyst formation with single medium. In the absence of any differences in overall performance, other factors should really be considered. And so if one considers these factors, I think one can conclude that single media is associated with some benefits. It is associated with a lower cost because fewer quality control assays are required, and a potential, importantly, for improved safety. There's one less medium to handle in the lab, and so there's less embryo handling, whether using the same media drop from day 1 to day 5, or whether the medium is refreshed. Even if it is refreshed, one presumably would not have to as rigorously rinse the embryo as you're transferring it from the first dish on day 3 to the second dish on day 3, which contains the same culture media. So now let's discuss blastocyst versus cleavage stage transfer. So the rationale for extending culture to the blastocyst stage involves consideration of activation of the embryonic genome, which we believe and was shown in the late 80s to occur late on day 3, in which the embryo itself starts to actually express its own genome. And because this is the case, and because this is required in order for blastocyst formation, the rationale for extended culture then rests on this premise, because not all embryos in the human develop satisfactorily to the blastocyst stage, because not all undergo embryonic activation. And so day 5 transfer or day 6 transfer, if one is performing extended culture then, allows self-selection of the morphologically best embryos, those embryos that have undergone embryonic genomic activation and which form blastocysts. So this is one of the primary tenets and rationales for extending culture to the blastocyst stage. In addition to that, more recent work in 2014 from Dagan-Wells group has shown that the aneuploidy rate of embryos is increased when assessed on day 3 as compared with day 5. The overall aneuploidy rate on day 3 was assessed to be 84%, and on day 5 it was assessed as being 56%. And each one of these columns here reflects the types, within each column reflects the type of abnormalities observed, and the 3 columns actually express the aneuploidy rates for good quality, average quality, and poor quality embryos. And as you can see here, the embryo stages for the blastocyst. So based on this analysis, culture to day 5 may allow for greater selection of euploid embryos, which might be another rationale for extending culture to the blastocyst stage. And then one mustn't forget the uterus, and there are potential uterine issues that might impact our justification for performing blastocyst culture. There has been work that has showed that there is improved synchrony between the embryonic stage and the uterine environment, and that there is actual disturbance due to elevated estradiol and progesterone, and so this improved synchrony may actually be of even more importance in that respect. In addition to that, there's at least one study, this one I'm citing here is from Fanchen et al. In 2001, that actually demonstrated that there was a reduced uterine contractility following blastocyst transfer as compared with cleavage stage transfer. And indeed, a further study, also by the same group, showed that there was a reduced risk of embryo expulsion when blastocysts were transferred. So the rationale for extending culture then can be summarized as follows. Self-selection of embryos results in higher quality embryos developing to the blastocyst stage, possibly a lower incidence of aneuploid embryos, and the self-selection is due to the fact that the embryonic genome is activated on day 3, and not all embryos undergo activation, and so not all embryos can proceed to the blastocyst stage. Secondly, the uterine environment may be more favorable for blastocyst transfer, and so therefore extended culture should enable transfer of fewer embryos of higher quality in a more receptive uterus. Together then, higher implantation rates and lower multiple rates should result following blastocyst transfer. So what does the evidence from the randomized control trials tell us? So this is the data that is tabulated from the Cochrane review that was updated in 2016. And what it shows here are the 5 different outcome variables that I pulled out of this Cochrane review that I thought were of particular importance and interest. And you can see that I've shown the number of trials that were involved in each analysis, the 2 different types of culture paradigm, transfer paradigm, day 5-6 versus the cleavage stage, and then on the right, the adjusted odds ratios. And as you can see, there are 3 different variables which actually showed a difference between the cleavage stage versus blastocyst. First of all, there was a significant increase in the transfer cancellation rate following day 5-6 transfer versus cleavage stage transfer. It was actually more than doubled in the blastocyst transfer arm. Secondly, and importantly, the live birth rate was actually significantly improved in the blastocyst arm compared with the cleavage stage arm, 39.1% versus 30.3%. And thirdly, with respect to embryo freezing, there was more embryo freezing, or more cycles, retrievals had embryo freezing in the cleavage arm than in the blastocyst arm, 62.4% versus 47.2%. Of note, the cumulative live birth rate was actually not different in this Cochrane review, although of note, there was an undefined number of cryoembryo transfers performed. And one might, I think, be justified in being a little bit surprised by this cumulative live birth rate, because more embryos were actually frozen following cleavage stage transfer than following blastocyst stage transfer. However, as I've mentioned, more robust embryos will be frozen if they are at the blastocyst stage because of the self-selection in culture. So the overall viability of those embryos frozen at the cleavage stage might be reduced compared to the viability of blastocysts frozen. So let's look at a little bit more depth at the cumulative live birth rate for cryoembryo transfers performed just within 1 year of retrieval. So this work was published in 2014. It involved single embryo transfers both for day 3 and also for day 5. And what you can see here is that overall, the cumulative live birth rate per initiated cycle was actually 48.5% following day 3 transfer and 48.0% following day 5 transfer. In other words, they were absolutely equal, so the clinical equipoise remains with respect to cumulative live birth rate, at least from this study, regarding blastocyst transfer versus cleavage stage transfer. A further analysis in this particular study was performed to actually ask what the time to pregnancy was for the cumulative live birth rate after a single fresh transfer and then cryoembryo transfers within the year of retrieval. And as you can see, as is shown in this graph, the time to pregnancy following fresh day 3 SCT followed by CETs versus fresh day 5 SCT following CETs, the time to pregnancy is about a year longer following the cleavage stage transfers versus the blastocyst transfers. In addition to the data I just presented, monozygotic twinning has also been assessed following day 3 versus day 5 transfer. And as is shown in this graph, you can see that blastocyst transfer is associated with an increased risk of monozygotic twinning in all the studies in which this has been analyzed. Nevertheless, I will say that there is some data that does show that the incidence of monozygotic twinning very strangely sort of occurs in waves, so that a lab might see a high incidence of monozygotic twinning and then none for a while and then a high incidence again. And the basis for this we really don't clearly understand. But nevertheless, at least based on the studies that have been looked at, there is an increased risk of monozygotic twinning following blastocyst transfer. And likewise, we performed a study which we published in 2008, where we showed there was also an increased risk of monochorionic twinning, which was particularly the case when ICSI was performed, but not so in the absence of ICSI in the day 5 embryo transfer group. So the summary of the clinical outcomes for cleavage versus blastocyst transfer are as follows. The day 5, 6 or blastocyst transfers are associated with an increase in live birth rate following fresh transfer, no difference in the multiple birth rate, an increase in monozygotic and monochorionic twinning rates, a decrease in the number of embryos frozen, no difference in cumulative live birth rate within 1 year of retrieval, and a shorter time to pregnancy, which I think of note likely has an emotional benefit to patients, and an increase in transfer cancellation rate in unselected patients, which is an important point. So furthermore, the evidence supporting the above is all of low or very low quality. In addition to the clinical outcomes that have been assessed, obstetrical risks have also been analyzed, and a meta-analysis, systematic review and meta-analysis that we published late last year revealed the following. First of all, that there is an increased risk of perinatal mortality with blastocyst transfer. There's an increased risk of preterm birth with blastocyst transfer. Here you can see the relative risks. Increased risk of very preterm birth, an increased risk for large for gestational age, but a decreased risk for small for gestational age. Now, importantly, the evidence for each of the above, just as with the other RCTs that I presented, is unfortunately of low or very low quality, and most of the absolute incidences with respect to obstetrical outcomes following blastocyst transfer versus cleavage transfer are actually very small. So the the clinical importance of these observations really remains to be assessed with scrutiny and rigorousness. So which patients should have extended culture? So in this presentation, we've not at all talked about or discussed pre-implantation genetic testing, but I think we all know that the accepted way to biopsy embryos these days is to perform it at the blastocyst stage and do trifecta biopsy. So as a result of that, at least with current protocols, all PGT patients, pre-implantation genetic testing patients, should have blastocyst or extended culture. So for the non-PGD patients, really a definitive answer remains to be determined as to whether there is benefit with blastocyst versus cleavage stage transfer if one considers the cumulative live birth rate. Appropriately powered randomized controlled trials with current technologies are required really to resolve this issue. Patients should be counseled regarding the pros and cons of each transfer day, and because of the risk of having no blastocyst to transfer, an algorithm for transfer day should be used. And so this slide actually just presents to you the algorithm that we use at Brigham and Women's Hospital for the prospective selection of day of embryo transfer, again for non-PGD patients. So at the fertilization check, if a patient is under 40 years old and has fewer than 6 zygotes, she automatically will have a day 3 embryo transfer. If she has at least 6 zygotes, she will have a day 5 embryo transfer, with the minor caveat that if her blastocyst conversion rate has been very poor on a previous cycle, we may do a day 3 transfer on her. If the patient is 40 or more and she has fewer than 8 zygotes, she will have a day 3 transfer. And if she has 8 or more zygotes, she will have a day 5 transfer, again with the caveat that I mentioned. If she's previously had poor blastocyst conversion, we may take her to a day 3 transfer instead. Day 3 is recommended, therefore, in our program with patients with a poor prognosis for blastocyst formation. Now, I will say that we have done work to show that a minimum of 4 zygotes is actually required in order for us to get at least 1 blastocyst to form overall on average in our patient population. But when we designed this algorithm, we decided to be a little bit conservative because we certainly didn't want to be in a situation where a patient had no blastocyst to transfer whatsoever. So this is the algorithm that we currently use at Brigham and Women's Hospital. So key points with respect to blastocyst versus cleavage stage transfer. Blastocyst transfer is associated with a shortened time to pregnancy. There is emotional value in this and reduced costs to patients. Selection of better prognosis patients to blastocyst transfer is recommended. A lab that offers extended culture must have an efficacious and reliable culture system which supports development of embryos to the blastocyst stage. It must have a proven vitrification protocol for blastocyst freezing, and it must obviously have adequate incubator space to keep all embryos safe. And then, as I mentioned, there are other considerations which must be taken into consideration. And particularly, extended culture does increase the cost to laboratories because of the increased time that embryos are in the incubators and the increase in culture media that is used if the embryo media is replaced or refreshed on day 3 for culture of the embryos out to day 5 or day 6. So final comments to sort of wrap this entire lecture up. So I think it's fair to say we've come a very long way since the birth of Louise Brown nearly 4 decades ago. We have greatly improved our understanding regarding the biology of human gametes and embryos and the development of the pre-implantation embryo itself. We have improved our understanding regarding the basic requirements in running an IVF laboratory with all of its complexities and in culturing embryos to the blastocyst stage. We've also made great advances in the protocols for ovarian stimulation and embryo transfer. And indeed, it's fair to say that more and more patients are leaving our clinics pregnant, which is absolutely fantastic. Nevertheless, I think we need to proceed with caution to ensure that all our technologies are appropriately validated before their standard implementation in the clinical IVF lab, And also, we need to continue to assess the outcome, not by pregnancy or delivery, but by birth of a healthy baby, preferably one single baby at a time. And so I'll just close by just actually reading the Barker hypothesis, because I think it's actually very powerful. A baby's nourishment before birth and during infancy, as manifest in patterns of fetal and infant growth, programs the development of risk factors such as raised blood pressure and glucose intolerance that are key determinants of coronary heart disease. However, I think that we really need to be looking further back from actually the gestational period and consider the male health and the female health for the product of spermatogenesis and oogenesis and the ovarian stimulation regimen and all the laboratory effects that might actually come into play as we're culturing our embryos in vitro, whether we culture them and transfer them on day 3 or we culture them on day 5. This is certainly not my idea alone. Many people are now much more concerned about how these influences might actually impact down the road on the development of the individual fetus, the baby, and then ultimately and very importantly, the development of the adult herself or himself. So thank you very much for your attention. I'll be delighted to try to answer any questions that you have. Thank you.